Welcome to Scigest, your fortnightly science from the Bioeconomy Science Institute. Kia ora koutou katoa. Welcome to Scigest. I'm your host, Hilary Ireland. Today on the Science Life for Me series, we have a very special guest, scientist and communicator extraordinaire of all things fungi, Dr Peter Buchanan. Peter has spent nearly 50 years studying New Zealand fungi and sharing his passion with the world. Now retired, Peter continues to share his knowledge and expertise as a research associate in the systematics team at the Bioeconomy Science Institute in Auckland. And because Peter's expertise and contributions are vast, we have a two-part series to cover it all. First, we get Peter's take on the fascinating world of fungi, and in the second part, we discuss Peter's research and impact. Peter, welcome to Scigest.
Thanks, Hilary. Nice to be here.
Good to have you. Your science career has spanned almost 50 years. Starting at the Department of Scientific and Industrial Research, the DSIR, through to Manaki Whenua Land Care Research, and then here today at the Bioeconomy Science Institute. Can you take us on a bit of a whistle-stop tour of the roles you've held over the years?
Well, I was really fortunate to be selected by DSIR back in those days, straight from university. In fact, at that time I was just about to finished my bachelor's degree, and the DSIR needed a scientist, so they asked the professor at the university, did he have anyone who was interested in fungi? He did have someone, he had me, and without an interview I was given an opportunity to work for the DSIR, and that entailed finishing my degrees, going overseas, being bonded to come back from overseas, which was not a hardship, considering there was a job waiting for me to continue with DSIR. And I was employed as a researcher, looking at fungi, mainly from the point of view of plant pathology. So those disease-causing fungi and fungi causing decay of wood. So research was the basis of my initial years with the DSIR. The opportunity then came to get into a sort of team leadership role and research leadership, and I took that, which certainly diminished the research aspects of my work, but I was working with wonderful people who were doing much more talented research than I was, and so I could encourage them and work along with them as well. So the staff relationship, the people management side of things took on a bigger role as time went on, but my passion for the fungi never waned, and I've remained a firm advocate for the science, and especially for the science relating to fungi. Nice.
By all accounts, you are a very passionate mycologist, a scientist specialising in the study of fungi. How did you initially become interested in mycology? What was it that grabbed your imagination?
Well, many people who have become mycologists often talk about being interested in fungi from a very young age, and that was not me. I was very, very interested in animals, and I had multiple fish tanks and kept frogs and lizards and all sorts of things as a child, and was determined to be a zoologist when I went to university. At university, I was absolutely stunned to realise that all through my education, a whole kingdom of life had been completely missed. So I went, as I said, intending to specialise in zoology. I was firstly surprised to find that plants could be interesting as well, and then I was basically overwhelmed to think of this new kingdom, the fungal kingdom, that I'd never actually even thought about before. And that realisation somehow has carried me through the decades. And I'm very keen to help other people realise this amazing world of the fungi, which most people don't seem to be aware of.
Indeed. I had a similar experience at university too, went in for environmental reasons.
Yes.
And then discovered the cellular and molecular biology.
Right.
And it was the same. It was like this whole world exists.
It just opens up. And it's a huge world. It's a world that I feel kind of, a bit sad that so many people don't know anything about it.
Yeah.
Because I've seen people similar to me who've sort of suddenly discovered it and have then just been really, really interested and passionate about it, just like me.
Well, let's dive into it a little. Before we venture into your research and impact, let's do a little quick dive into the world of fungi. So yes, one of the key things I've come across is that many people confuse fungi with plants.
Yes.
But as you say, it's its own kingdom. So where does the fungi kingdom sit in relation to all of the other organisms, and what's its size and scale? And
Well, yeah, as you said, Hilary, I, like you, I basically grew up thinking that fungi was some kind of plant. I think that was what I was told in my education pre-university. And more recently, I think a lot of people think that they're microorganisms. So I've had a bit of a soapbox mission in my life to try to dispel this idea of fungi being something else than they are, and also more recently especially to dispel the idea that fungi are microorganisms, because that's the way the education system has been treating them in New Zealand. So they are, scientifically, the second largest kingdom of life on this planet. And so we typically talk about plants and animals and think that that covers more or less biodiversity. It's so far from it that it grates on me, and always has for the last many, many decades. And fungi are a separate kingdom. They are the second largest kingdom of life. Only the animals are ahead, and people, when you say animals, they probably think of the vertebrates, but of course the vertebrates are a tiny group, and it's the insects that are and other invertebrates are the main groups of animals. So the animals are the biggest kingdom, the fungi is the second biggest kingdom, and the third is the plants. And plants are so much smaller in terms of number of species than the fungi. But it's taken humanity a long time to realise that, and hence I'll offend botanists by saying most of the plants are already known, and very, very few of the fungi are known yet, despite the work that's gone in. So it was an underdog for me, and suddenly I was able to do research on this underdog kingdom to try to redress the balance and try to also change this idea of flora and fauna, which people like to use if they're thinking they're a bit above saying plants and animals, flora and fauna. And then I've always championed the missing F word.
Yes.
And the missing F word is fungi.
Nice.
And so just instead of the duplicate we need a triplicate. It's flora, fauna and fungi.
That's cool. What are some of the examples of fungal species that we might interact with on a sort of regular basis but are commonly mistaken for other elements of life?
Well, one that comes up all the time is lichens. People can often distinguish a lichen and they think that they are looking at a plant. And sure, they're often green, they're mostly green, but the bulk of the organism is a fungus. It just happens to be living in association with green algal cells, and that's why it looks green. And the algae are producing food for the fungus, and the fungus is giving the alga a home. So the name of the lichen will be the fungus, and the algae are a much smaller proportion. But people look at it and it's green, it must be a plant. No, it's a fungus. It's primarily a fungus. I think people probably can, when they see organisms, they probably understand that they are fungi, but the thing is most people don't see them. I mean, they see moulds and they will probably know that yeast is a fungus. They might know that yeast is a fungus. So they're using, and we're consuming bread and wine and beer, and we know that yeast is involved there. So we know fungi are involved somewhere there. We know that our food goes mouldy. We know that we need to ventilate our homes to stop too much mould in them, and that spores are bad and that. And then, come autumn, they will see mushrooms coming up, and yes, there's the fungi again. But the realisation that the fungi are there all year round, almost everywhere they look, is something that people find, I think, very hard to understand. That fungi are within the fallen leaves, they're within the fallen wood. They are the reason that we have nutrient cycling happening that converts all of the fallen wood and leaves and trees and dead bodies of animals, etc., converts all of that to the soil as nutrients. The decomposition process, which we absolutely take for granted, and we see rotten wood and we just think it's, how is it rotting? Oh, it just rots. It only rots because fungi are living in it. So dead wood is really alive. It's a huge complexity of different species all fighting to feed on that dead plant material, and hence returning the nutrients to the soil. It's a magnificent system that we just take for granted, and it's the fungi that do it. So the fungi are there doing all this stuff, but they're not producing any grand output that people can often see. And so it's what they do that I think is incredibly important, that I like to help people understand. And then there are big fungi that people can see. They produce large reproductive structures, the mushrooms and the brackets and all that sort of thing, which is why they're not microorganisms. The largest organism in the world is a fungus. So microorganisms are the bacteria and other unicellular animals, plants and fungi. That's the microorganisms. Microorganisms are small things you can't see. Fungi you can see, not necessarily in every stage of the life cycle, but they are not microorganisms. They're not unicellular, most of them, apart from the yeasts. So it's this idea of pigeonholing fungi into some sort of thing that people might relate to is not the way to go, because fungi are unique. They are a separate kingdom. So yeah, that's part of my soapbox, to just lift fungi out of this obscurity into an understanding for people of what they are and what they do. Other things, though, it's sort of in the more sort of economic sense. We grow all the different crops in New Zealand. Almost all of our crops are introduced plants. And almost all of those plants, those crops, have diseases. And most of those diseases are caused by fungi. So you could say, oh, fungi are bad. Well, yeah, those fungi are. Most of them are introduced fungi. Our native fungi don't typically cause diseases of any of our crop plants. They might naturally cause some diseases of native plants, but there it's in balance, because evolution has basically enabled things to live together. But we go and put crop plants from overseas, and in bringing them into New Zealand in the old days, of course, we brought their diseases in with them. So we've got exotic introduced fungal pathogens parasitizing introduced plants that are crop plants. And that includes grass as well, of course. So it's important that Aotearoa New Zealand understands the diseases that we have here. And so plant pathology is a really, really important part of our whole economy and our whole pastoral and farming primary industry system, which is where I started at university, because in those days there was a chair of plant pathology. And that was a recognition of the importance of plant disease for the viability of the New Zealand economy. It still is a really important component. It just doesn't have the profile now, it seems, that it used to have. But that's where I started in a project to look at a disease-causing fungus on a grass in Northland. And that was paspalum grass. And that paspalum grass was valued by farmers in those days for their summer feed for their cows and sheep. This disease had come in, and my project was the start of my PhD, in fact, was to study what was causing this disease and what impact was it having on the pasture, and ultimately could it be controlled. There is an irony in that link with paspalum grass for my initial study. I am now in a voluntary basis on a trust looking at how can we control and eliminate paspalum grass from a pristine nature reserve, that being the island of Aotearoa, Little Barrier Island, where paspalum is a threat as an invasive grass in a native ecosystem. So I've gone from trying to preserve paspalum to trying to kill it over a few decades.
Can you use the same fungus?
The fungus will be there naturally. It's not enough to kill.
Okay.
And it turned out that the dairy farmers didn't have too much to worry about anyway.
Oh well, good. It's good to research it anyway.
Yes.
You touched on so many great things there. I have to touch on a couple of those just to learn more. The largest organism is a fungus?
Yes. There's a statement, the humongous fungus among us, which relates to a study of a fungus called Armillaria, I think Ostoyi, in America. America always has the biggest of everything, of course. And they were able to study this fungus, which a single colony of it, which had grown over decades, I think, from a point source radiating out, as fungi do, and that they could identify the same genetic individual that was all of these many, many years old and had covered many square kilometres of ground, growing through the soil and onto various trees and continuing to grow out. And if you take the mass of all of the underground fine threads or hyphae of the fungus and the fruit bodies and everything, you had an organism much larger than any blue whale. So
Wow.
You've got the largest organism being a fungus. Again, a microorganism? I don't think so.
Does the hyphae networks or the hyphal networks, mycelium networks, is that the same
Yes,
thing?
that's it.
Do they go out like ripples on a pond and die in the centre, or is it living all throughout, like that largest organism? Was it alive everywhere?
I think it does. It is moving out, but it depends on where the food base is, and the food base is as it radiates out. The fungus will keep feeding as long as there's food there. So if it actually, as it expands out in this circular expanding colony, if it comes upon major food sources, that will then be a focus for its growth.
Right.
Beyond. And it's like in a stump of a tree, a fungus can grow on that stump, or many fungi will be growing in the stump for as long as the resource is there for the food of the actual wood cell walls to provide food for the fungi. So, yeah, you have these major sort of food sources that the fungus latches onto and continues growing with still retained growth where the food is.
Gotcha.
Yeah. And when you mentioned, you reminded me also of the sort of the underground fungi. The other sort of major importance of fungi is their relationship with the roots of plants. The plant fungal association is almost ubiquitous around the globe, and I think over 90% of plants naturally have fungi growing with their roots, and could not grow as well as they do without those fungi. So this so-called mycorrhizal relationship is really fundamental to life on Earth. And again, we typically take it for granted. But the way I've had it explained to me in a sort of general sense is the plants used to be in the sea, and their movement onto the land occurred about 400 million years ago. The fungi were already present on the land, and quite large structures, almost tree-like, of the way the fungi formed in those days. And so the association between fungi and plants began at the very point of plants moving onto the land and becoming terrestrial. So the plant root and the fungal associates with the root enable the root to tap a much larger volume of soil, and therefore minerals and water come to the plant from these fungal hyphae that are associated with the plant, but radiate out into the soil far beyond where the roots could reach. And in return, the fungus, of course, needs to get something for this relationship. The fungus gets the carbohydrates that the plant produces in the leaves, which go down to the roots. Some of those then feed to the fungus. So this plant root fungal association, you can see how it's beneficial to plants, and it's so widespread. And again, it's another thing we just take for granted. Oh, the plants are growing well. Have you thought about the fungi that are actually helping them to grow well? It's just to help people realize that life is actually really complex.
Yes.
But it's complex for a reason. And those relationships between the roots and the fungi are just so important.
So you've talked a lot about the roles of fungi being decomposers, and then there's all these symbiotic relationships, ones with plants, the mycorrhizal associations, and then lichen in that symbiotic relationship with algae. Do they perform other roles in the ecosystems that they're in that's sort of outside of decomposition?
Well, outside of decomposition, you've got the symbioses that you mentioned, and you've got the parasitism as well, which is the disease-causing organisms. So I like to think of it as two beneficial functions, the decomposition and the symbiosis, mycorrhizae and lichens, versus the negative side, which is there are parasitic species that kill living plants, and I guess you could say kill living animals as well. And that's the negative side. But it's the negative side that I think gets accentuated far too much, because most fungi are not damaging to people, which is what people mainly react to. I guess fungi also, though, could be seen because they do, in that they decay naturally in the forest, they also decay food products that we produce. So that's a negative as well. And New Zealand has to spend a lot of effort, as it does, just in the plant pathology of the growing plant, but it also has to do a lot of work to protect the actual product, the fruit, for example, from decomposition, from attack by the fungi. I'll say again, though, that the fungi that are the problem are typically the introduced fungi that came in from overseas when we brought the crop in. It's a pity that we don't have sort of native fruit in New Zealand that we can make a lot of money off, and then the native fungi would probably not be as much of a problem. I haven't really thought much about that, but I suspect that might be the case. And there's good reason. I mean, it's the science that I've been doing and my colleagues have been doing gives the foundation of the understanding that we need to actually get the best yield from our crops, but also to get the best storage of them so that we can actually trade with them and export and all the rest. It's all understanding the fungi the specific fungi that are involved and how they work. It's not as if I can say, fungi are all good. Of course not, they're not. But I can say that it's so important to understand the fungi, because then we can address the issues that we need to control those that need control.
Indeed. And
to get better value out of the products that we need for our economy.
Indeed. Yes. So as a mycologist, part of your role is to identify and characterise. But with fungi, most of the organism is hidden from view. Occasionally you might see a mushroom or a toadstool.
Can we address that?
Well, yeah, let's address that.
It's another soapbox of mine. Years ago, involved in the taste panels for the early cultivation of shiitake mushrooms. Shiitake mushrooms then were not known to most Western people, and when they were taste tested, people were asked what they would call them, and the word toadstool was mentioned, and I found that incredibly offensive, because shiitake at that stage, and possibly still is, the second most important cultivated mushroom in the world, and the word toadstool has no relation typically to an edible mushroom. I had also had a bit of a mission with our child at that stage, and subsequent other children, to try to reduce the use of any negative term for the fungi. And to me, toadstool was quite an offensive term, because it suggested something that you should steer away from.
Gotcha.
It's also, I guess, used in children's books, and that's sort of that's almost acceptable. But anyway, in our household, it was the T word, and we called things mushrooms, whether they were edible, poisonous, beautiful, not beautiful, didn't matter. They're all mushrooms. We don't need this word, the T word.
The T word.
And looking across the other kingdoms, we have no word for poisonous animals, and we have no word for poisonous plants. And I think people typically think of toadstools as poisonous mushrooms. So the T word it is.
It is out.
And it's transferring through to the next generation because certainly my first daughter has taken it on board and is training her daughter likewise.
Yeah. I'll have to check in with my own children. I think they're probably more influenced by the Disney element of it, that it's just a thing for a toad to sit on.
Yeah, well, that's kind of a nice, pretty
but still,
image too.
It's not a toadstool.
No.
It's a mushroom.
That's right. Yep.
So mushrooms are obviously very distinctive, but how do you go about going into the wild and identifying and characterising fungi when they're not in bloom, I guess, in autumn? What are the distinctive features of a fungi to a trained eye?
What are the distinctive features of a fungus to the trained eye?
Sorry. Whoa.
That's another soapbox. Fungus,
Yes.
singular, fungi, plural. Plurals are used quite often, but without recognition of the singular. But that's all right. Hilary, don't be embarrassed about that. That's
I won't.
entirely fine, and I'm used to it. But I still react to it. What do I look at if I'm going collecting? I mean, I'm typically looking for what I can readily see with the eye, but I probably will need to look a bit more closely. Children have a big advantage in looking for fungi because they are closer to the ground. So for me, I will typically be turning over things, turning over logs, looking on the underside. Fungi produce their reproductive structures as opposed to their feeding stage. The feeding stage is often hidden in the wood or in the soil. But they produce their reproductive structures not for our edification, but just to produce spores. But they produce them in this vast variety of different forms. And the ones that I was particularly interested in are those that grow on wood. So I would be turning over logs to look on the underside because that's the side where there's the most moisture typically. And fungi, as well as needing food, they also need moisture. So the underside of logs is a wonderful place to find fungi, and that's where I would typically be looking. And why would I be looking? Well, yes, I want to identify what they are, but I actually want to take samples that can be put in the national collections. Now, that I've always worked throughout my life in the National Collection of Fungi, and it's not just one collection, it's two collections. And the collections they could be thought of a little bit like museums, but they're actually research museums. The whole foundation of our understanding of organisms is based on real physical examples of those organisms. So the national collections are a really important part of our foundational science, where we actually get together examples, real examples, in the case of fungi, dried fungal specimens that document these organisms. And we also have all of the information about where they were found and when, by who, and in association with what plant. These collections are really fundamental. They're the evidence base. So when we say we have this particular organism in New Zealand, we can go to physical material to confirm that. It's not just something written in a book somewhere. It's actually physical material. And our whole science is based on accurate identification of organisms and their interaction with other accurately identified organisms. With the fungi, it's even more kind of complex than that because we have this collection of the dried fungi, the New Zealand Fungarium, but we also have a living collection. And so when I go and look in wood, for example, for a fungus, I might not be able to see the fruit body, but that doesn't mean the fungus is not there. And so how do I actually go about looking for the fungus or showing the fungus is there? You can take a piece of wood with a fungus inside and you can grow it on an agar medium. And then you can actually produce this colony of the fungus. It's a feeding stage of the fungus. It's not the reproductive stage that you see as a fruit body. It's the feeding stage. And you can grow that on agar. And then in the National Culture Collection of Fungi, this living collection of fungi, you can actually freeze the fungus, eventually putting it under liquid nitrogen, and have it alive for decades. It's amazing with fungi. You freeze it down to minus 196, the temperature of liquid nitrogen. You hold it there in little drinking straws that we buy from Pack and Save, would you believe? The scientific product that actually we need to keep for decades in frozen liquid nitrogen, or in the vapour of it, has come from Pack and Save. And we have 22,000 strains of fungi, and we have bacteria as well, in this natural culture collection. And we can warm them up whenever we need them to do research by putting them in 30-degree water. And they go from minus 190 whatever to 30 degrees, and we put them on agar and they start growing again.
Wow.
I mean, fungi are incredibly interesting in just being able to do that.
Yes.
But that enables our research to cover not just the dried material that we have as our evidence base, but the evidence base also of the living material that we can actually store in frozen condition for decades.
And how have modern technologies helped in the study of fungi?
Yeah, well, the way I described it, I guess, with fungal specimens and with cultures, we certainly in my early days, we would primarily use the microscope to look at the fine features of the fruit bodies, how the spores are produced, all of the dimensions of the different components of the fruit body, and we would look at how the fungus grows in culture. But the revolution that came from molecular biology and being able to get particular components of the DNA sequenced, and then to be able to basically use this massive international database of sequences of all forms of life that have been discovered and sequences produced in this international gene bank, we can then get the sequence of a particular fungus, a diagnostic part of that DNA, and blast it to the international database and get back a response of the likely name of the organism that we're observing. And that can be done increasingly at a cheaper price and more rapidly. That doesn't supplant the use of the microscope and our visual observation of things, but it is an amazingly objective way of getting an identification, and it points us in the right direction anyway, even if we don't get an actual match with something that's already been sequenced before. And mostly because of the diversity of fungi, you're not getting an exact match.
No, right.
Because the New Zealand species, if it hasn't been studied in New Zealand, is very likely to be different from overseas. So the molecular revolution was huge for mycology, and it meant a massive change of our classification because it showed up relationships that we never realised. Just because things look alike doesn't mean they are evolutionarily related.
Indeed.
And our taxonomy, our understanding of the classification of fungi must be based on evolution, not just on what looks like something else.
How do you define a new species? What features go into...
Yeah, well, a friend of mine, Bevan Ware, has a statement, fungi don't carry passports. And nor does any organism. But it seems easier with animals and plants, although I'm probably being naive, to tell where one species finishes and another one starts. It's always subjective to an extent. But at least with DNA, we can put some measurements on it and look at similarities between entities and have sort of benchmarks that you can choose to follow as to how different something needs to be before it is described as a new species. But in the older days, we used to do it by mating studies.
I was wondering if you used those.
Yeah, and that was a lot of fun because you had to actually, you either had to get a single spore from several examples of what you were studying and try to mate them to each other to see if there was compatibility, so-called compatibility. And that produced all sorts of challenging results and didn't always make sense, in fact. Yeah. And more recently, we've discovered that some species actually can interbreed. And that evolution, of course, is an ongoing process. So there are species that are very closely related and may not have actually eliminated the possibility of being compatible with one another, but they may be geographically separated, and so the spores never come into contact, or some other reason that maintains their separation, and they may be diverging, in fact. In fact, everything is either, I guess, changing as we look at it. Its evolutionary time, though, is a very, very long, has a very, very long scale, as opposed to our just short lifetimes as we observe it from year to year. So, yeah, molecular biology has been massive for mycology.
Is there any other new technologies on the near horizon that you think will transform mycology?
Certainly the AI and increased use of computing, which is part of the whole use of the whole sequencing side, I guess. That's only going to make things easier, but it has been, I think, misinterpreted by people to suggest that it's going to replace the need for humans to be involved in the process. But life is incredibly complex, and I don't think AI will have an ability to... It can more rapidly interpret what is already known,
exactly,
but it has trouble finding things that aren't known.
Yes.
And AI can't observe. Anyway, I don't have a full understanding of AI. I just know that it will be incredibly useful for our science, and it will work hand in hand with the expertise that we need from our scientists in future.
Indeed, yeah. It's been great speaking with you today, Peter. Thanks so much for sharing your thoughts on the fabulous world of fungi. In part 2, we'll talk to you again about your research impact and thoughts on the future. But for now, we say goodbye. Thanks for being here.
It's been a pleasure, Hilary. Thanks very much.
And thank you out there for listening. If you'd like to know more about the topics covered on Scigest, check out our episode pages on the Plant and Food Research website. Please also subscribe to or follow the Scigest channel and rate and review each episode. Why not give it 5 stars, particularly this one? Thank you and mā te wā.